looking at this idea of replication, that kind of 5-step process that all viruses seem to go through. It doesn't matter which type they are, they have to attach. They have to penetrate into the host cell, either the nucleic acid or the whole thing. Then they go through the replication process, and then the maturation or assembly stage, and then release. So when you think about the different viruses, you have, when we say virus, we usually think about those that are infecting animals. And then of course, the bacteriophages are the ones that infect bacteria. And so it depends on which virus or phage you're looking at and how they're gonna go through, especially the release stage. So I'll talk a little bit more about that. But as you can see from this visual, some viral particles are pretty straightforward and kind of bare bones, and others are pretty fancy. And so if you have something as complex as some of these phages, like T2 or T4, got a lot of proteins to make in order to build that. And so it'll take a little bit longer in that regard. But basically, when you look at, so this is T4 phage, which only infects bacteria, and they have kind of mapped it out, like, what's going on in terms of from start to finish. And basically, I put this up here just to show you kind of the step-by-step process in a timeline. And then, because this one's a little bit more complicated, you have a lot more proteins that have to be made. So the transcription and translation will be a little bit more lengthy, and just to get them to get this all done. And then of course, assembly is going to take a fair bit too, to assemble all those parts correctly and get it so that it's a fully infectious particle ready to go. But you can see, even though it's fairly complicated in its structure, it from, at least when they do lab-based experiments, about 25 minutes start to finish. So that's pretty fast. It's pretty impressive that you can get these particles made that quickly, considering their complexity in design. So if you think about some of these viruses or phages, we've already talked about this way back when we looked at how horizontal gene transfer is going on, and that sometimes you can get the viral DNA pasting into the host DNA. And so that's the lysogenic pathway. And so you're going to be needing to know the difference between lysogenic versus lytic and what's going on with that. And so with the lysogenic, when we were looking at horizontal gene transfer, we said that this is a way for the second host to actually get potentially some bacteria that the virus might have accidentally, so to speak, grabbed from the first host when it was making more of itself. But if we just think about the virus now and not horizontal gene transfer, yes, their DNA, their double-stranded DNA can sometimes integrate into the host bacterial DNA. And in essence, it's just kind of a passive, passively hiding out in that form, because now when that bacteria goes through binary fission, all the progeny cells will have the viral DNA as well. And so it's kind of a nice lazy way for the phage to go ahead and get replicated into the next generation of that host bacteria. And so they still don't know all the details of how this happens, but they know that there are certain key proteins that the phage has to have in order to keep its DNA pasted into the host DNA and just stable and just stays there for a while. And so, and then at least in lab experiments, the way that they can get that viral DNA to get active is to stress the cell in some way. And so usually in a lab, you hit it with UV light, that's pretty stressful, or you can add a chemical that's a mutagen, and that will trigger the virus DNA to get cut out of the host DNA, and then it usually switches immediately into the lytic pathway, which is typically bad news for the phage, because, for the host cell, because that's when the phage decides to, so to speak, take the cell hostage and make more of me, and then they'll bust out of the host cell, typically killing the host. microbe. So we looked at this a while ago. This was during the horizontal gene transfer. And so they're showing the lysogenic pathway off to the right. And so here you can see it's the dark green phage DNA is starting to paste in. And so then once the phage DNA is in the host DNA, we say it's a prophage. And that can get into the offspring, into the progeny cells now when that cell goes through binary fission. So it's a nice, easy way to spread through the population just by staying stable in the host DNA like that. And then you hit it with ultraviolet light or somehow stress the host, and you can see they're shifting now into the lytic pathway. And that usually ends up with making, all the phage particles, and then the cell typically dies. So kind of a slick little system there to be able to infect many progeny cells through the prophage system. This is another diagram showing basically what we just went through so this is good to study off of because it has some of the explanatory text on here. But comparing the lysogenic pathway off to the right again of getting pasted into the host DNA versus the lytic. The lytic is kind of straight down here again, okay? So when they study this in the lab, most phages that they've studied tend to cause cell lysis to the host. So when they release, it tends to happen where all the particles are like, let me out all at once, and the cell will bust open. There's so much damage done to the host cell that it lyses and breaks open. But there are some viruses who do a slow release process. And so that's what's being shown here, where especially with viruses that are very petite, here's a there's a filamentous, there you go. I was trying to think of the word. They call this a filamentous shaped virus. And so it's very skinny. And so when it releases, it can do it in a non-damaging way because it's so small. And it can kind of more slowly, they don't all bust out all at once. And because of that, the host stays intact and you don't destroy the host cell. And so in fact, the host bacteria can actually keep on doing some of its own stuff. Like it can slowly replicate itself. So it can do some of its own activities, even though it's infected with a virus. And here they're showing how the virus gets out, but the host cell is still alive. It's still doing what it needs to do. It just does it slower because it's in an infected state. And so they often say it extrudes out or it has a slow release mechanism. And the host stays alive. So I guess if you were a bacteria, you'd prefer to get infected by something like this because at least you can stay alive even though you're infected. Okay, so the release step can be pretty brutal for the host, or it can be not so bad, and the host can actually stay alive. Switching into the animal world, though, you get some differences when you look at the viruses that infect animals, including us. When you compare them to the phages aren't enveloped typically, but animal viruses often do have an envelope that surrounds them. Remember, that's just a bilayer membrane that was stolen from the host cell. And so with animal viruses, they have a little bit more complications because, animal cells are a little bit more complex. They have the nucleus and all the organelles. You have different locations of where DNA is, or the nucleic acid is made, where transcription is done, in the nucleus, and then translation is out in the cytoplasm. So you have all of that to deal with. And so all the nuclear activities, you have the DNA, getting transcribed to RNA. You have DNA replication going on in there too. then the maturation of the messenger RNA, where they have to process the RNA a little bit, all that is done in the nucleus. And then you have to get all the messenger RNA out to the cytoplasm in order to do your translation. And so the viral, has to adhere to these rules. When you get into a eukaryotic cell, you have to work with what you have on hand. And if a eukaryotic cell is doing this, well, that's what you have to do as a virus too. Bless you. So here, it's just a visual, just trying to show, that you have non-enveloped or naked viruses versus enveloped. And then, they can be RNA or DNA, it doesn't really matter. You have options there. But coming back to this idea, especially when you think about animals in general, because they're multicellular, they're complex, they have different tissues and organs, typically. So that goes back to that idea I mentioned last week, that viruses tend to have specificity for what types of tissue or cells they will be able to infect based on the receptor proteins of the host. And so examples, Ebola has a pretty broad tropism, so to speak. That just means that it can get into a variety of different host tissue types. That's why it's such a deadly disease. It is very fast acting because it's very broad in what it can lock onto and attach to, which then allows the virus to gain entry. And so Ebola can kill within 48 hours. And so it is mainly to do with this idea that it can attack so many different tissue types of the host. On the other hand, the papillomavirus really only can attack epithelial tissues with the correct receptors on them. And so there's a wide range of different viruses that kind of like narrow spectrum and broad spectrum antibiotics. You have these different viruses, but another difference we tend to see is that animal viruses tend to enter in intact. They don't just like inject their DNA. They tend to come in intact. Whereas phages, they tend to just inject their nucleic acid. And so they'll, when virologists use the word virions, they're referring to the entire viral particle. And then once the virus gets in, they'll get uncoated, which just means you have to break apart the capsid so that you can release the nucleic acid. Okay. So when you think about what types of viruses infect animals, the difficulty from the viral perspective, it depends on, is it a DNA or RNA virus? Is it single-stranded or double-stranded? And so if they're a double-stranded DNA virus, then it's pretty straightforward because that's what the animal cell is dealing with. You have double-stranded DNA to start with. And so they can immediately, take advantage of the replication machinery, and it's pretty straightforward. RNA viruses, on the other hand, it's more challenging, of course, because they have to get some DNA made, make their RNA, or they can use their RNA-dependent RNA polymerases to get their RNA. So the main point here is that DNA viruses have it the simplest when you think about infecting animal cells. RNA viruses, depending on what type they are, if they're retroviruses too, those are RNA viruses. Some of those have, they'll use reverse transcriptase. And so it just depends what virus you're thinking of in terms of how complicated is this going to be for them to go through their 5-step process and replicate more of them and deal with the logistics too of an animal cell, a eukaryotic cell. So they have to deal with the ribosomes that the host cell has. And so their RNA has to work with that. And of course, that's all happening in the cytoplasm because that's where translation occurs in animal cells. And then I'm going to show you a couple pictures of how viruses do this, because once they've gone and made copies of their nucleic acid, they've done transcription and translation, then they have to do all that assembly. And of course, if some stuff's still stuck in the nucleus and some stuff is in the cytoplasm, all of those parts have to find each other in order to assemble into a full virus that can be infectious. And so I'll show you a couple options of how some viruses do that. We'll look at that in a second. And then as they start to finish up their assembly, and usually it's during the release step that they'll, if they're an enveloped virus, that's when they will steal part of the host membrane and wrap it around themselves so that they can, it's a really good way to hide out from the immune system of animals. Typically, they do modify the membrane a little bit. They'll put their own proteins in there so that it's good to go for infecting the correct receptors for attachment. So typically, it depends on the virus. Sometimes the viral particles, as they bust out of the animal cell, it's going to be a complete lysis. And so if you have herpes, like cold sores, that's the lysis that's occurring, where you get the sores. Budding, on the other hand, is a more controlled passage through the host membrane. And often in that process, they'll grab some of the membrane and wrap it around themselves. Now, depending on the virus, they might do it through the cell membrane and wrap the cell membrane around themselves, or they might take some of the endoplasmic reticulum membrane. So it might be from an internal organelle. It's not always the host cell membrane. So it just depends which virus you're thinking of, which membrane material they're going to grab. But regardless, it is host membrane. And so it's a really nice way to hide yourself. Yes.
Is budding always making an envelope or some of the time?
Not really. I hear budding being used in a more slow release process as well, where they don't kill the cell. So I don't take it as only stealing membrane material when they bud, but they definitely say that word budding when they are wrapping. It just depends how they're phrasing it and what they're talking about. So here, the papillomavirus, which is a double-stranded DNA virus. So just trying to show the complexity that it has to deal with, in the nucleus, you have to make more copies of the double-stranded viral DNA. Plus, you have to do transcription to take the DNA to the messenger RNA phase. You have to get the messenger RNA ready to go so it can work with the ribosomes out in the cytoplasm. And so once they're working, once the ribosome and the messenger RNA hook onto each other, then you have a translation. So now they're showing all the proteins that are getting made, and they're showing that the proteins go back in to the nucleus where everything else is there from the virus, all the bits and pieces of the nucleic acid. And so they put it all together in the nucleus and then they come back out. So it's this process of sometimes, you have certain things happening in different spots, but they all have to come together somewhere in order to assemble. And then you can see the exit out. Here with herpes virus, again, double-stranded DNA virus. Again, just showing that it depends, they're trying to show that the membrane material can be taken from a cell membrane or organelle membrane. You have to do all the assembly where all the pieces are all in one spot. So again, it's typically back in the nucleus. And then here with HIV, this is an RNA, single-stranded RNA virus. And so you have that kind of complexity because you have to take that RNA and make double-stranded DNA off of it, because the host machinery, that's what it has to work with. It has double-stranded DNA, RNA, and protein. And so the virus has to get that situation as well. So you have a reverse transcriptase that's going to make DNA off of the RNA. Very unusual. And then here, it looks like they're getting the assembly occurring out in the cytoplasm in this case. You don't have to know which virus does what. It's just important that you realize that there's logistics that the virus has to deal with in terms of all the steps of replication when you deal with a more complicated animal cell like this. Okay, so we've talked about phages that attack bacteria, animal viruses, and then plant viruses are kind of a different situation. They don't really have the 5 steps like what I just talked about with the phages in animals, where there's attachment and penetration and replication, assembly and release, because plants have that really thick, tough cell wall. And they've yet to discover a virus that can penetrate through that. And so what tends to happen is that you have other mechanisms that have to come into play first in order to get the virus into the plant. So can you think of one way a virus could get in? If it can't get in itself, what could it do to get in? It can't directly just like inject its DNA into a healthy plant cell. We have yet to see that happen. Already wounded, exactly. So if there's a wound where the virus can get in that way, definitely. If any of you are working with plants in the greenhouse and there's some issues with other things in the greenhouse, can you think of another method that they could get into the plant? Pests. Yeah. So if you think about some insects that can very precisely, like aphids that have a very precise stylet mouthpiece that they can pierce into the plant. And if that aphid is carrying the virus, that's a great way for the virus to get in. And so when you have insect pest infestations in the greenhouse or wherever, you have to think about that. Not only is the damage direct from the pest a problem, but what viruses are they bringing with them in their saliva that they're injecting into the plant? So it can be a real double whammy. And so mechanical transmission is the idea that you typically have a wound where they can get in, or an insect typically. And then in the seed, unfortunately, sometimes there are viruses that make it into the developing seed of the mother plant. So as the seed is forming, the virus gets in there, and then the virus finishes maturing and getting ready to go, and it's in there. You can't like get rid of it. And so that's a problem too. So it's a little different, and it's primarily because the cell walls of the plant are just so protective. These are some ideas when you think about plant viral infection. So I believe A is called a potyvirus. It's filamentous. But the insect here is trying to show that you have that kind of transmission. Figure C with that tulip, the striped tulip. So that was a huge, people like paid enormous sums back in the Victorian era for these types of tulips. They actually called it tulip mania. It was a real thing. And they didn't know it was caused by a virus, but a virus causes that. And so because of that, it was inconsistent. So if you happen to end up with some striped tulips, then you could, charge a pretty penny from that, the mother plant producing, offspring with the virus infected into the seed like that. But it was like this mania. People were like manic over it. And I read some of the things that they would charge, people could charge to have people buy this. And it was ridiculously high sums. And then this is not so pretty, but you can see symptoms like this showing up on, I think this is a peach fruit. And so it can be very challenging to tell the difference between a viral infection on a plant and like a mineral deficiency. It can be really tricky. It's not easy to tell apart very often. But anyways, when you look at plants, so this diagram is trying to show that really nice thick cell wall, okay, all that gray. And that just makes it pretty impossible for the virus. And so, but if a aphid injects its mouthpiece into the cell, deposits the virus, then the virus can get from cell to cell to cell through the plasmodesmata. And so once it's in, then it can go throughout the whole plant. And so I would like you to know that term, the plasmodesma, is singular. Plasmodesmata, if you put a TA on the end, that's plural. So the plasmodesmata is like an internal tunnel system linking neighboring cells together. And once the virus gets in, it can just go through that route very easily. It's tiny enough, it can easily fit through the plasmodesmata and go from cell to cell to cell. It can also go through the vascular tissue, which is analogous to the blood vessel system in a human or animal. And so once they're in, then they can really cause some problems, but the hard part is getting in. All right, so they're a little different. Here's a specific plant viral infection going on here with the columbovirus. And again, plants are eukaryotic, so you got to deal with the nucleus and all the organelles and different places for transcription and translation. But then they're showing once it's done, it can then start going into nearby cells. So each viral particle can just spread throughout the plant and cause more problems, more infection. Now, luckily, we have very sophisticated immune system. And so we definitely there's certain mutations that can make certain individuals more resistant to certain viruses. They've documented that with HIV. We have very healthy immune systems typically, as long as you take good care of yourself. And so you have both an innate and adaptive arms to the immune system that can be very useful to fighting off all kinds of viruses. And they've been looking more and more at this RNA interference idea that a lot of organisms have, even down into the archaea, where basically RNA interference is just saying that you basically destroy the viral RNA. So they're looking more at that. And on the upside, they're looking at using phages as therapy delivery systems. And so there are some, it's not like we're at the total mercy of viruses. However, viruses do mutate so quickly that even if, we have certain types of defenses, they can often mutate to get around it. So, but the phage therapy, you're probably going to hear a lot more about that in the next decade or so. I think that's going to become much more of a thing. But when you think about it, really, from a viral perspective, this is a good quote: an inefficient virus kills its host, a clever virus stays with it. If you are a virus and you kill your host, your mandate as a virus is to spread and make more of yourself. I mean, that's what virologists thinks, that's what a virus is supposed to do. It's supposed to spread. Can't really spread if your host is buried or cremated. So it doesn't really make sense from that perspective to kill your host. So often what happens is if a virus is particularly virulent and does kill the host, it's usually because the virus and the host haven't had a chance to co-evolve with each other and come to a more stable relationship. And so like with the COVID epidemic we just went through in the last few years, the idea that after the first year or so, the virus should have become less harmful, which it did. And then our immune system became a little bit better at fighting it off, which it did. And so we unfortunately got to witness this coevolutionary process of having the disease not nearly as fatal. If you compare the data to, from this year to back in 2020 when it all started. So
Which reminds me, I'm gonna post a homework assignment that I'd like you to do. It's gonna be through EduCat. And it's looking at the idea of using the process or thinking about the ideas of evolution and ecology in order to make pathogens less virulent. And so it's a TED Talk that I want you to watch and then answer a series of questions on it, okay? But I think it's a really good talk, and I wish more medical doctors would think about this idea of how to, force a pathogen to be less virulent. I think if we could do that, really ramp that up and really go for it, we could make highly pathogenic viruses and other pathogens thing of the past, or at least very unlikely, very small population. So I'll be posting that in EduCat, yes. When's it due? It's gonna be a while out because you have the exam to think about first. So I'll put it after the exam due date. Oh, and by the way, there's another TED Talk I posted on Dr. Bassler on quorum sensing, so make sure you watch that too. Okay, I'm going to get into the second slide set now on viruses, part 2, which should be posted. And I wanted to also point out, there's a couple of readings in EduCat that are available now. And so this is a New York Times title to an article, "Trillions upon trillions of viruses fall from the sky each day." Now, ordinary people might freak out about that, but in this class, hopefully by the end of this content, you'll realize this is no big thing. It's not anything to worry about. But of course, being journalists, they like to shock and awe people, so they put that in there. So it's listed as viruses in our atmosphere. So it's a pretty short article. There's a couple of good key points that are being made in there. And so please read through that because I'll be asking at least one question on that in the exam. And then there's a very small, short PDF. I think it's just 1 page maybe, on the origin of viruses, where they get into some thoughts on that. And I'll be asking about this on the exam too. But basically, there's 3 big ideas on how viruses might have evolved. And so please know those, the very basics of them. Okay, so those are 2 short readings posted in EduCat. All right, so this slide set's basically getting at some of the ecology of viruses. And this is where I want to try to get across the point that most viruses could care less about us, and they're actually doing good things for our ecosystems and habitats. So just like when we talk about the human microbiome, when we talk about all the bacteria and fungi and viruses that hang out with us. So they've kind of copied that word. The virome is all the viruses in a certain ecosystem. They'll use the word virome. And it can be, don't forget about the viruses that kind of hide out in the host cell that have pasted their DNA into the host DNA. So you have a prophage, if it's a bacterial virus that's doing that, or a provirus is the term that we give if it's an animal virus that has inserted its DNA into the animal DNA. Then we call it a provirus, right? So it just sits in there. The DNA of the virus or the phage sits in the host DNA and can get passively transmitted to the next generation in that way. Then if you take this a step further and you say, what if that viral DNA never really does cut out from the host DNA? Then we start using the term endogenous viruses, where it's a provirus or a prophage, and it just never it's just like permanently in there now. It's become part of the chromosome, and it never seems to get activated. And sometimes that can actually be good for the host. It means the host will not go through a lytic phase. The virus won't kick into the lytic phase. And maybe some of those genes over time could be useful to the host. And so um, the classic example they point to is that the cyanobacteria, the genus Nostoc, can fix nitrogen, which is very unusual. And they see that it has gained that ability because of a prophage that is endogenous now. You have it stuck and it's just persisted over evolutionary time. What's even weirder to think about, you talk to a virologist, they'll say as much as half the human genome may have arisen from viruses or virus-like elements called transposons. So if you talk to a virologist, they will say that viruses are incredibly important in the evolutionary process for all organisms on Earth, including humans. And so I'll mention a little bit more about proviruses and what they've done for us specifically in a minute here, but looking at viruses, so that New York Times article, trillions upon trillions are falling down from the sky. You have viruses in any environment you look in, any ecosystem, no matter how harsh it may seem from our perspective, there's viruses everywhere. And they are definitely playing a role in the functioning of that ecosystem and in the evolutionary process of life on Earth. 1 × 10⁷ viral particles per milliliter of seawater. So pretty abundant in the ocean as well. And they've been studying that pretty carefully. They've gotten some pretty nice data now looking at how important viruses are in recycling of nutrients. Because if they break open cells during the lytic phase, they are releasing a lot of nutrients. So they're trying to show that. They often refer to it as the viral shunt. You don't need to know that, but like you talk to a virologist, that's how they'll describe it. But basically, they're just saying that viruses are playing a role in that nutrient recycling. And so they're looking at that process. And so this is basically showing the same thing, a little bit pared down. But you see here, they're looking at the organic matter, it's getting broken down. If you have cells lysing open and breaking open, you have all that breakdown going on. And then, of course, you have the food web where you have grazers that will be taking that up. And then those that feed on the grazers and on and on and on. So they really do play important roles. Now they're looking more carefully at the carbon cycling going on, and they are realizing that viruses really do play a major role in that compared to what they knew beforehand. So viruses are everywhere. And if viruses are, kicking into the lytic pathway and they're breaking down and killing off host cells, that's not always a bad thing when you look at certain ecosystems, because otherwise you could get imbalances of what species are in that ecosystem. And so acute viruses are the type, that's the term given to the ones that are pretty much always in the lytic pathway, they're always doing that. And they can limit population densities and help keep things more in balance. And so they're looking at this idea that you have to factor them in when you're looking at all the ecology, the ecological interactions that are going on in these different ecosystems. And they're really starting to look at this more with the human and the microbiome of the gut. And they know that there's all kinds of viruses in everyone's gut, and that they are playing a role in what microbes are there, what population levels are they at in the gut microbiome. So definitely a lot of interest in that is gaining, especially now that they understand a little bit more about the general human microbiome. So here is just some visuals talking about the different things that microbes are doing. You can see on the figure C, on the lower image, they're showing a before and after with the virus. So the algae, the algal bloom, harmful algal bloom going on. And then the virus is multiplying and going through the lytic pathway, killing all those algae. And so from that perspective, it's a very nice visual showing that viruses can have a huge impact on population sizes in these different ecosystems. So we know that viruses are a tool to get nucleic acid moved, DNA moved. So we talked about transduction when we looked at how viruses can participate in horizontal gene transfer. We know that that can happen. The horizontal gene transfer, now they're suspecting nanotubes are also doing that. So it's not just viruses, and also extracellular vesicles. So there's more than one way to get gene transfer, but viruses are a big player. All right, so remember to think about those ideas. And so I'll mention a few things about how certain endogenous virus in our chromosomes have given us certain traits that have been considered definitely a benefit. But before we get into that, there's this paper published in Science. That journal, by the way, is like one of the, premier journals. If you get published in Science, it's a big deal. So the title is "A Virus in a Fungus in a Plant. Three-way symbiosis required for thermal tolerance." So certain plants can tolerate really high temps. And when they were asking, how is a plant doing this? They discovered an endophytic fungus. And so they thought it was the fungus. But wait, there's more. Then they realized the virus has to be in the fungus in order for this the heat tolerance to work. And so that was surprising. So you need to have the virus inside the fungus and that inside the plant in order for the plant to survive. And so this is the virus that they isolated in this study. And here they're doing a greenhouse study where they were working with the tomato plant. And if it doesn't have, so this is the tomato with the fungus, but no virus. So they cured the fungus of the virus. So it was just the plant and the fungus, and it died at high temps. Then they put the virus, made sure the fungus had the virus, and the fungus got into the plant, hit it with high heat, no problem. So again, this idea that you need to have, a third entity involved in a virus. I mean, if you weren't looking for this, not everyone has an electron microscope where you can easily look for viral particles. So often if you see inconsistent results in your experiments, one thing you should keep in mind with these types of studies is, well, maybe there's a virus at play in here and we just aren't seeing that because we weren't testing about it. We weren't thinking about it. So this is a mock-up trying to show no fungus, the plant dies at high heat. If the fungus is infected with the virus, then the plant survives. So tricky little system here. So again, showing that this plant definitely benefits not only with the presence of the endophytic fungus, but having the virus there too. So they can do good things for other organisms. They're not always killing and lysing their hosts. All right, so let's think a little bit about the endogenous viruses where they can remain long-term, the DNA can remain long-term in the host. So this is a table where they've documented this type of situation. And so Clostridium, I think we did mention this briefly with horizontal gene transfer. So Clostridium botulinum, if it doesn't have the correct prophage in it, it won't cause botulism disease. So you could get exposed to it and it's no problem. But if the phage inserted its DNA with toxin gene on it, well, then it becomes virulent. From Clostridium's perspective, having that virulence is a good way to get access to nutrients. So, and the other thing too about Clostridium, that bacteria, it doesn't need to infect a human to survive. So botulism can kill humans. It's one of the deadliest toxins biologically made that we know of. But Clostridium doesn't need to do that. That's why this remains so deadly. Because Clostridium, it doesn't need to infect a human. It's a soil bacterium. So when you get this type of situation, it can be very deadly for the human. There isn't this co-evolutionary process.
going on, because the bacteria doesn't have to have access to the host nutrition. It's happy to live in the soil. Knows how to get its nutrition, and its needs met from what's going on in the soil. So this is a particularly nasty situation for any human that gets exposed to the Clostridium that has a prophage for the toxin. You can see a few other ideas here in the bacterial world, looking at often their toxins. On the left, on the right in the pink box here, now we're getting into what they've discovered in humans. And so human endogenous retrovirus, so we're thinking about proviruses that are endogenous viruses. They were a provirus, and then over evolutionary time, they just became permanent viral stretches in the chromosome. And a lot of the work has been done where they show that the placenta never would have evolved if it weren't for viral DNA. They point to the viral DNA in the chromosomes of the human in order for secure placental and maturation and formation of a placental tissue. Placental tissue is pretty darn important. That's how you get into the next generation. And so it's a huge aspect of the human life cycle. So again, just showing that these viruses have, a profound impact on their hosts and on the evolutionary process. So they're continuing to look. What else? Some of these endogenous viruses that are in our chromosome, but we have a lot of endogenous viral DNA in our chromosome, a lot. And so this is a little, you can use this for studying, but basically looking at what are they doing for us? Depending on which virologist you talk to, between 40 and 80% of the human genome may be linked to ancient viral invasions. So again, just pointing to the idea that viruses have been there since the beginning, and through the evolutionary process, they're interacting with all these organisms and influencing their evolutionary process. And it's kind of weird to think that so much of our DNA is viral. That's the origin of it. And yet through the evolutionary process, it makes us who we are. So it's kind of freaky to think about, but all right, we'll stop there and continue on. And then Wednesday, we'll finish up viruses and start on the human microbe interactions.